Good morning and good afternoon. Welcome to the complement system webinar today, hosted by ALPCO in partnership with Diagnostica. My name is Terry Fisher and I'll be the webinar moderator today. Before we get started, I just wanted to mention a few items. Due to the number of attendees we have today, we will hold all questions to the end, but you're free to submit them anytime throughout the webinar in the questions section on the toolbar. We'll do our best to answer all the questions during the session, but if we're not able to get to any, we'll certainly follow up on any of the outstanding questions after the session. Without further ado, I'd like to introduce our speaker today. Dr. Jörgen Wieslander currently serves as the Chief Scientific Advisor for Diagnostica. He's held various university teaching positions, founded the VIS Lab, a full-service testing lab in Sweden, and has over 170 publications to his credit. So without further delay, I will turn over the session to Dr. Wieslander. So Dr. Wieslander, the floor is yours.
Thank you very much, Terry, for this nice introduction. So today I was going to speak about the complement system and give some background on principles, some interesting diseases, and then describe the functional assays that we have available at Diagnostica. As you all know, the innate immunity is an important part of our defense system. So we have the adaptive immunity, which is composed of cells like T cells and B cells that can adapt and learn, and we have the innate immunity, which is a ready-made defense system composed of circulating proteins like complement and different kinds of cells like phagocytic cells and neutrophils. The difference between the systems is that the innate immunity is ready-made present in the germline, while adaptive immunity is generated through somatic recombination. So the innate immunity has a limited number of receptors of pattern recognition molecules, like 100 to 1,000, while the adaptive immunity has an enormous array of possibilities to generate. The drawback with the adaptive immunity is that it takes time, while innate immunity is there from the beginning and can act directly on infections. So what is then complement? Complement is one of the major parts of the innate immunity. It is composed of more than 40 different proteins. It is like a triggered enzyme cascade that leads to recruitment of inflammatory cells, opsonization of pathogens, and killing of pathogens. So it is a very powerful system that needs to be under control. As you see from this slide, there is a lot of complement proteins in the blood. For example, C3, which is one of the central molecules, there is 1 gram per liter. C4, there is 0.12 to 0.3 grams, factor B, 0.6 to 0.44. C1 inhibitor, a little less, but there's an enormous amount of these proteins in the blood. The proteins are produced by many cell types. It can be hepatocytes, monocytes, macrophages, endothelial cells, epithelial cells, fibroblasts. But the liver is the major factory for complement proteins. So if you have a liver problem, there's always a risk that you have a limited amount of complement available to defend you. The name complement was established already more than 100 years ago when Ehrlich and Morgenroth did an experiment where they took colibacteria and mixed with antibodies to them, and that have no effect. Then they took colibacteria and human serum, and there was still no effect. But when they mixed colibacteria, antibodies, and human serum, the bacteria were killed. So the conclusion was that there is something in serum that works as a complement to the antibodies. As I said, The complement was discovered already around 1900. The alternative pathway around 1950, lectin pathway with MBL 1990, and with lectin pathway with Ficolins around 2000. So the three major investigators discovering the complement system was Jules Bordet, George Nuttall, and Paul Ehrlich. Two of them got the Nobel Prize. They defined the complement as a heat-labile component of normal human serum that is involved in the antimicrobial defense. Then in 1954 there was a publication on the properdin system that a scientist called Louis Pillemar had found. He was criticized a lot for this publication, and people didn't believe him really. And some say that he killed himself because of all the criticism he got from this publication. But it only took about 4 years later, then there was a new publication on an alternative mechanism for the properdin system. And then this was accepted, and it is now the alternative pathway of complement. Then it took some years until the lectins were found. So the first was the mannose binding lectin that was binding to mannose structures, and this is now the initiator of the lectin pathway. Then a couple of years later, in 2000 around, the Ficolins were detected. There's a couple of Ficolins. They are very similar to Ficolins, and MBL, very similar structure like tulips. Ficolins are more activated by glucosamine containing surfaces, while MBL is more mannose type of surfaces. So let's take an overview of the complement system and how it works. As I said before, C3 is a major molecule. Everything has to go through C3. C3 can have two paths. It can be split into C3A, which is a small molecule, and C3B, which is a large molecule. C3A is a signaling molecule that calls for help or attracts mast cells and neutrophils to the site of infection. C3B can have different functions. It can be IC3B and C3B on surfaces that will call for phagocytosis and label cells for opsonization. Or it can lead to the membrane attack complex, the terminal complement complex, that will kill. So the three major functions of complement is to call for help, to call for eat, and kill. The split between C3 and C3A and B can be performed in different ways. One is the alternative pathway of complement, that is surfaces with LPS, like on different bacteria, or it can be a spontaneous activation, and that probably takes place all the time in the body. So it's like when you have the car on idle before a red light stop, that there's still some activity all the time going on. To activate the alternative pathway, you need factor B, D, and properdin. Then there will be an enzyme that will cleave C3. Another way is to generate this cleaving enzyme from the classical pathway. So the classical pathway is activated by surfaces of IgG or IgM together with C1q RNS, forming the C3 convertase and cleaving the C3. Or from the lectin pathway, reacting with carbohydrates like MBL, Ficolin, with mannose or glucosamine structures together with the MASP enzymes that will form the enzyme and split the C3. So the complement is activated through different surfaces on bacteria, fungi, cells, on antibodies bound to surfaces, and which results in killing and phagocytosis of cells. So this is a more real picture of how it looks like. As you see, it's a complex network of different reactions. And if you look at this picture, there is yellow molecules here also. These yellow molecules are inhibitors. So almost all important molecules in the complement cascade has an inhibitor. So it can be circulating inhibitors or it can be tissue-bound inhibitors that stop the reaction on cell surfaces. So to remind you again that the major functions is anaphylatoxin, recruitment of inflammatory cells, opsonization of pathogens, removal of immune complexes, and killing of pathogens and cells. So if we look at the small split products like C3A, C4A, C5A, they will all lead to increase in the vascular permeability of blood cell or vessels, and this increased leakage will result in that proteins from the blood, like antibodies, like complement proteins, will leak out into the surrounding tissue, and then the polymorphonucleosides or lymphocytes will migrate out and start to help to kill the infection. And as I said, that the split will result in a small molecule C3A recruiting phagocytes, but the large C3 part, C3b, will immediately bind to a nearby surfaces like a bacterium. And a bacterium that has a C3b bound to it will be a signal for neutrophils to eat it immediately. So it will remove the bacteria. Another major function is to remove immune complexes. So while there is an antigen-antibody complex, complement can be activated, C3b is bound, and then red blood cells have receptors that will recognize C3b on immune complexes and take the immune complex to the macrophages that will take care of the immune complex. The red blood cell is released and can go into circulation again. So this is a powerful way to protect us against immune complex deposition in tissues, and this is probably a reason why complement system is involved in diseases like lupus, since you have a lot of immune complexes there. And then if we continue down, I said the third function of complement was the membrane attack complex. So if we continue down the line, here we have the C1q bound to antibodies. We have the C3 splitting C4, C5, C6, and then the chain of events from C6, C7, C8, and C9. So C9 then forms a pore complex, making a hole in the cell membrane. This will kill the bacteria. This is how it looks like on the cell surface. There are a lot of holes in the membrane, or from the side view you can see the pore formed so the proteins from the cell will leak out. There are many different surfaces that can initiate complement reactions. As I said, immune complexes are important, but also apoptotic cells, viruses, gram-negative bacteria, CRP bound to a ligand. For the lectin part, it is mostly microbes with terminal mannose or glucnac structures. For the alternative pathway, it can be many different surfaces like the bacteria, fungi, viruses, tumor cells, etc. So this is a short summary of the system. We have the classical pathway, the lectin pathway, and the alternative pathway. The classical pathway is initiated by anti-antibody complexes. The lectin pathway by lectin binding to carbohydrates. Alternative pathway by different pathogen surfaces or self-activation. The complement is activated and the cells are recruited. Opsonization of pathogens, killing of pathogens will take place. So why should we then analyze for complement? Because there are complement deficiencies, and they are associated with certain diseases. This is some examples of complement deficiency and disease. So this first is from a 4-year-old boy with a complete C4 deficiency and systemic lupus erythematosus. The second is from a woman with factor D deficiency and meningococcal infection. So a lot of the deficiencies will lead to increased sensitivity to infections or autoimmune diseases. So this is a list of deficiencies that has been determined, and if you look at the classical pathway, it's a general tendency that deficiency in classical pathway leads to SLE, glomerulonephritis, and some to infections. MBL is the most common deficiency in the Western world, where it can be up to 1 to 10 that is deficient in this, perhaps more. If we look at the alternative pathway, most of the deficiencies lead to Neisseria infection or meningococcal disease, but also could be some lupus-like disease. The membrane attack complex, C5 to C9, deficiencies in that will lead to Neisseria infections. Deficiencies in the regulatory proteins like C1 inhibitor will result in hereditary angioedemas, a typical hemolytic uremic syndrome, age-related macular degeneration. And deficiencies in the membrane-bound regulatory proteins, which can give infections, but also a disease like paroxysmal nocturnal hemoglobinuria. So what do you do when you find deficiencies in a small child that have a lot of infections? You can vaccinate, because when you vaccinate you strengthen the adaptive immunity, so you get antibodies to the bacteria that can help fight the infection. So for example, properdin deficiency, vaccination with the meningococcal vaccine is strongly recommended. If you have C5 to C9 deficiencies, vaccination improves, opsonization is recommended. C2 deficiencies, you can also vaccinate. Another aspect of complement analysis is that because complement is involved in many autoimmune diseases and activity reflects ongoing disease processes, especially in diseases like SLE. And as you can see from this picture, the red line is the clinical activity scoring. So at time 4 months, this patient has a flare of disease and is active. And at the same time, the blue line indicates that the classical pathway of complement is consumed. And as you see on the green line, the terminal complex is increased. So there's consumption of complement when there's a flare of disease. And this can be used to predict flares. As you see from this slide, that on the left side you see mild SLE, then there is no complement consumption. If you have intermediate disease, there's more complement consumption, and in the severe lupus glomerulonephritis, almost all patients have complement consumption already before they have the flare of disease. So the third aspect of complement analysis reflects therapy. So if you have a deficiency, you can give the deficient molecule to the patient. But also there's more and more interest in biologicals, that is monoclonal antibodies that can interfere and stop different aspects of the complement system. So for example, if you have a C1 inhibitor deficiency, C1 inhibitor inhibits C1R and S as well as MASP1 and 2. So it leads to classical pathway activation. But it also leads to angioedema that can be either acquired or inherited. But actually it's not the loss of the complement regulation that is the major cause of disease, but the lack of inhibition of the factor 12A and kallikrein of the contact system. So you can give these patients C1 inhibitor and successfully treat them. There's also trials ongoing to give this to other patients with sepsis and transplantation. As I said, the most common deficiency is the MBL deficiency. It can occur up to 20 to 30% in Europe. But MBL deficiency by itself is not enough to cause disease. But if you have an MBL deficiency, you have more, you can get the infections more easily. And if you have immunosuppressive therapy, like cancer patients undergoing chemotherapy, it can be important to know that you have an MBL deficiency because you have a higher risk of getting infections. MBL therapy has been tried in cystic fibrosis, for example, where patients often have severe lung infections, and those with MBL deficiency have a shorter life expectancy because they cannot handle these kind of infections. Then there's a lot of interest in finding drugs that can stop complement in different ways. So you can, for example, think about drugs stopping properdin, you can stop alternative pathway, stop C1q, and then you can stop classical pathway. The one that is used right now a lot is anti-C5, where you stop the terminal complex in forming. But still, you still have the C3a and C3b, so you still have attraction of neutrophils and opsonization, but not the killing of the C5b to C9 complex. This drug is used in paroxysmal nocturnal hemoglobinuria, which is a deficiency of cell surface proteins like DAF and CD59 that protects against complement activation on cell surfaces. And they get complement activation during night when pH is lower. So anti-C5 monoclonal antibody inhibits the MAC formation. There's also trials ongoing for other diseases like lupus and membranoproliferative glomerulonephritis. So this slide shows just how efficient the anti-C5 is in inhibiting complement function. So you can see before, all three pathways works very well, but when the patient is giving anti-C5, it completely blocks all three pathways. So measuring the complement function is very important when you give drugs, because you can follow the efficiency and the completeness of the complement inhibition. So then I will turn into analysis of complement. What methods are available? So there are several factors that influence the amount of components in the blood. There is a synthesis. As I said, most complement proteins are synthesized in the liver. So if you have a liver problem, you will get lower amounts. Complement is also an acute phase reactions to infections, so most components increase when you have an infection. And then there is in vivo activation, for example, autoimmune diseases where consumption leads to decreased amounts. So it is a sum of these factors that determines the level. So there are many methods you can use. There is methods for single components like ELISA, nephelometry, most commonly for C3, C4, but also other components can be measured like that. We have a biological activity and a classical hemolytic assays using erythrocytes, CH50, HH50, and now there's also ELISAs for classical, alternative, and lectin pathways. So the hemolytic assays has been used for a long time. And the CH50 is then the classical pathway measurement. And then you use sheep erythrocytes coated with rabbit antibodies. You add the serum from a patient and you have calcium, magnesium in the buffer. Then there will be lysis of the red blood cell. For the alternative pathway test, you have rabbit erythrocytes. You mix with serum and you remove calcium magnesium from the buffer. You then get lysis of the red blood cell. This is a classical test, but you need to have erythrocytes, and it's a bit cumbersome to standardize. Then we have the individual assays, like assays for C3 and C4, but you must remember that that's not enough to understand the complementary results. Look at these two patients. They have the same low level of C3 and C4, so you could believe that they have the same problem. But when we analyze for complement split products like C3DG, you can see that patient number 2 has a very high level of C3DG and also a very high level of the terminal complex, indicating that he has an active disease with active complement consumption, while the left patient has a low level because of liver failure. So the test that I will describe now was developed in a European complement group, and there were persons from different countries of Europe, from Norway, Sweden, England, Italy, Austria, Germany, Denmark, Netherlands, and Sweden. So the idea was to design ELISA for all the pathways of the complement system. So the ELISA uses specific coatings for each pathway. So for the classical pathway, there is IgM coated to the plastic. For the lectin pathway, there is mannose for the MBL and glucosamine for the Ficolins, and then there is LPS for the alternative pathway. This specific surface is combined with specific buffers for each pathway, making sure that only one pathway can run in this buffer. Then there's activation at 37 degrees, and then there's a common detection system by a monoclonal antibody recognizing a specific neoepitope on the C9 molecule formed in this terminal complex. So this is actually how it looks like in the wells of the ELISA plate. So you really build up the whole sequence chain of events. So for example, in the classical pathway, we have the IgM in the bottom, C1q binding, then C4, C2, C3, 5, 6, 7, 8, 9, and then the antibody to the C9 neoepitope, that is the detection system. So if something is wrong in this chain, there will be no signal in this well. And then you can combine the results from the different assays. So for example, if a patient has a deficiency in the properdin molecule, then there will be no signal in the alternative pathway, but the other will work well. And then you can, of course, design tables like this and find out what molecules can be the problem in the complement system. So for example, if you have a low level of a classical pathway and the other pathways are okay, the most likelihood is that it's C1q or S that is the problem. And in the same way, if this alternative pathway that is down, it must be properdin, factor D, or perhaps factor B. If MBL is down, it must be MBL. And then all the others. So if all is down, it must be the terminal pathway with all these different proteins, because that's common to all the complement pathways. Remember that MBL deficiency is very common, and that can be combined with any other deficiency. So this is an example of two patients with C1q deficiency. As you see that they have a very low level of signal in the classical pathway, but normal activity in the alternative and lectin pathway. This is an example of a C7 and C8 deficiency. And as you see, they are involved in all the pathways, so all pathways are down for these deficiencies. So some practical aspects. The buffers used are specific for the actual pathway, and you cannot exchange them during the assays. And there's different dilutions for samples of the different pathways. The activation step of 37 degrees is critical and must be timed, because the kinetics of the reactions are slightly different, so it's very critical to have a time this reaction. It's also important to store samples correctly, because complement activity is sensitive to in vitro activation. So heat inactivated serum or serum stored for a long time or serum that repeatedly has been freeze stored should not be used. Also, serum containing immune complexes or cold agglutinins can have low values. So if you find that all three pathways are low or that you suspect a deficiency, you should always ask for a new sample and retest it. So it should be serum, blood without anticoagulants that allow to clot for 1 to 2 hours. Serum should be stored at room temperature until frozen at -70 degrees. The maximum time before freezing is 48 hours, 24 is optimal. The sample should be analyzed the same day in the laboratory or frozen at -70 degrees. You should avoid freezing and thawing. So how do you follow up a test result? If you get a very low result in one pathway, you suspect a deficiency, and then you get a new sample and confirm the result. You can then further add the suspected protein or mix with deficient serum and redo the assay to clarify what protein it is that is deficient. If you get an intermediate result, it's a disease with complement activation, and you should check for complement activation product like TCC or C3DG, or you can have a normal result, of course. So there are several indications where you should screen for complement deficiency. That is, if you have a susceptibility to infections, if in all ages, if you have invasive meningococcal or gonococcal infections, especially children, if you have invasive infection with encapsulated bacteria, all ages, if you have repeated invasive infection with encapsulated bacteria, if you study systemic autoimmune diseases like investigation or suspicion of SLE, kidney disease, investigation of glomerulonephritis, if you have suspicion of angioedemas, hemolytic anemias, or atypical hemolytic uremic syndromes. So I would like to summarize then that complement is a major part of the innate immunity. It has three major pathways, the classical, lectin, and alternative pathway. Now there's an ELISA for the biological activity of the three pathways available. The ELISA can be used for screening of the major deficiencies. The classical pathway has a good correlation to CH50 and can replace the CH50 test. The alternative pathway has a good correlation to AH50 and can replace it. And properdin deficiencies are also detected. They were not detected in the old hemolytic assay. MBL is the first functional assay for MBL pathway, and Ficolin is the first functional assay for the Ficolin pathway. So the complement functional test can be used when you look for deficiencies. The result is that you have an increased risk of infections. When you have activation of complement system, when you, for example, follow disease activity like lupus, if you have patients having immunosuppression, because they need to have a full functional innate immunity, so it could be in transplantation, cancer therapy, autoimmune diseases, or when you have biological treatments where you try to inhibit one of the components of the complement system. Thank you very much.
Thank you, Dr. Wieslander. Great presentation. We'll take a look and open it up to questions here. See if we have any submitted yet. We do have a question. How does TCC analysis work?
You have an antibody for example, C8 or C6 that will and then you have the same antibody as we have in our test, that antibody to C9 neoepitope. So you coat with the antibody to C8 and you detect with an antibody to the C9 neoepitope, for example. That's a common way to do it.
Okay, thank you. Are there any other questions at this time? Let's see. Okay. Okay, on the slide you showed for the AP, why do you remove calcium and magnesium? I thought you would remove only calcium since magnesium is necessary for the assembly of the APC3 convertase.
Yeah, it's. I think it's correct that it's enough to remove calcium. Yes.
Okay. And there's one follow-up to the TCC question, just asking more specifically what you're measuring, a soluble form of TCC.
Yes, it's a soluble form of TCC you measure.
All right, and there's just one more question. It's pertaining to the presentation. If the slide deck and recording will be available, we are planning to archive the presentation and recording, and we'll make it available to the attendees in the following days. Any other questions at this time? Okay. If not, I will say thank you to Dr. Wieslander for taking the time out of his day to give the great presentation. And here is my contact information. If there's any additional questions or comments in the coming days, please feel free to email me with those. Again, thank you, Dr. Wieslander, and thank you, everyone, for attending today.